We're in Chapter 2, Part 6. Last video lecture for Chapter 6. It's the one we've all been waiting for. Just kidding. Well, sort of. We still need to cover proteins and nucleic acids. On to the proteins. Proteins are the third of the four types of biomolecules that we're going to talk about. In comparison to both the lipids and the carbohydrates, we are talking about a molecule that now has not only carbon, hydrogen, and oxygen, but also has nitrogen as a key component. Sometimes sulfur will make an appearance, particular amino acids, not all of them, but sulfur is important as we'll discuss in this section. Proteins, you probably already know, play a functional role in a cell, including as support structures, as enzymes, in regulating things that are going on in the cell, helping with homeostasis. Proteins transport components around the cell and across the membrane of the cell, and of course also play a role in the defense and offense of an organism. Amino acids are the building blocks, and they're composed of a backbone of nitrogen, carbon, carbon. Hopefully you can see that in this purple box down here. We have the nitrogen atom and we have a carbon atom and another carbon atom. That's the backbone. Then, as we're looking at the functional groups we discussed before, we might recognize that within the amino acid, we have both an amino group, and that's shown in that orangey-like box, as well as the carboxyl group over here in this yellow box. Did you get that question I posed in the earlier video correct? Did you recognize the carboxyl group on the opposite end of the amino acid from the amino group? So we have an amino group at one end of the amino acid and a carboxyl group on the other end. The other important component on an amino acid is the R group. That's shown here in that white box. An R group can be different things. They're different groupings of atoms that make one amino acid different than the other. In humans, we have 20 common amino acids. In microorganisms, some will use an additional amino acid. The R groups are what defines the amino acids as specific amino acids. The backbone is the same, the R groups differ. Those side groups, those R groups, will affect how the amino acids interact with one another and also how a protein interacts with other molecules. This R group that is shown in this particular set of examples, this kind of aqua color, we can see the R group can be different things, right? The simplest amino acid has hydrogen as its R group. That's glycine. We discussed it earlier. But we can have things like the large ring structures as seen in phenylalanine, or the sulfhydryl group in cysteine. This is an unusual amino acids that humans don't use, but bacteria sometimes do, selenocysteine. Here's glutamine with its branch structure, and this is serine. These are just a few examples of the different amino acids that are present in life. Importantly, peptide bonds are the name of covalent bonds that form between amino acids. Hence, proteins are sometimes called polypeptides because of the numerous peptide bonds that are occurring in a protein. And this joining of amino acids is no different than the other types of dehydration synthesis that we have already looked at, but because it's happening in such a key molecule, it's given its own special name, a peptide bond. You can see here that the hydroxyl group of one amino acid and the hydrogen of the other amino acid is going to be removed in order to remove water in order to form that covalent bond between the two. This is known as a peptide bond. Proteins are going to take on an ultimate final shape, and that shape is sometimes referred to as form or conformation, and it's important for function. Without the particular shape or form, the protein will not do its job. It cannot function like it's supposed to unless it has folded up correctly into the right shape, the right conformation. While we look at these different images, we're going to explore the organization of proteins. Organization of protein covers 4 stages. So the first stage, depicted here over on the upper left, is showing amino acids linked together. That's the primary structure. The primary structure consists of the amino acids linked together in a specific order. Any given protein has a specific order of amino acids. That's the primary structure. It does not change, or we have a mutant form of the protein. The secondary structure can take on one of 2 different forms, and the same protein can have regions of both. One of the secondary structures takes on an appearance of a helix, called an alpha helix. You can see here red dots. Hopefully this is familiar. The dots indicate that we have hydrogen bonds formed between the side groups, those R groups, in the amino acid. These bonds cause the amino acid chain to coil up into a helical form. That's one type of secondary structure. The other type is more like corrugated cardboard, where we have this fold going back and forth. That's called a beta-pleated sheet. Those are secondary structures, either alpha helix or beta-pleated sheet. The hydrogen bonds between the R groups, that's what's holding the shape in place. The tertiary structure are those regions folding up on themselves. You can see in this example in this blue and green, where this long amino acid chain took on a secondary structure in different regions and then kind of folded up on itself into a glob, which is sometimes referred to as a globular structure. In some proteins, this is the end. It will now do its job. That's tertiary structure. Some proteins stop here, but not all. That's an important concept. In some proteins, you will not get functionality until 2 or more folded subunits come together. You need at least 2 tertiary structures. The final amount is determined by the protein you're dealing with. In this case here, you're seeing a quaternary structure where we have 2 subunits. One is shown in blue and green, and the other is the more peachy red color. Those 2 tertiary structure subunits have to interact in order to get a functional protein. When they interact to give you functionality, that's quaternary structure. In some, quaternary structure is when you need to have multiple subunits of folded tertiary structure polypeptide chains interacting to give you a functional protein molecule. Make sure you understand these levels of structure. Make sure that you can explain it and answer questions about the structures of protein and what points you achieve, at what points you achieve that functional protein. Of note, proteins can be more than just amino acids. Within a cell, there will be enzymes that can add things to proteins. You may hear terms like glycoprotein, meaning sugars have been added, or nucleoproteins, where nucleic acids get added, or lipoproteins, where lipids get added. These are proteins that have had things added to them after they're formed in the cell. These are variations of protein molecules in a cell. The addition of components to the protein allow the protein to do a specific job or gain a specific functionality in the cell. Our last macromolecule is nucleic acids, and the subunits are called nucleotides. I need to draw your attention to the image at the bottom. Note that it is labeled ATP. ATP, in fact, is a nucleotide. A lot of times students have never realized this until we begin to talk about it. ATP is an RNA nucleotide because, as you can see, the sugar is labeled ribose, which is a component of RNA. DNA has deoxyribose. So what are the 3 components of a nucleotide? Well, one is that 5-carbon sugar, ribose or deoxyribose. You can see that the carbons are labeled here in blue. So we have 1 at this position, 2 here, 3 here, 4, and 5 on this kind of like arm waving up on the side. You'll notice that there is oxygen as part of that ring. The points that are chemical shorthand, where you assume a carbon and hydrogens exist, you'll see that there's a base attached to the carbon in position 1. And that base can be one of 4 different things. When comparing RNA and DNA, 3 of those bases are the same, G, A, C. In RNA, we might have a uracil, U, as the 4th, and in DNA, we might have a thymine, T. We'll come back to those in a little bit. This is the base at position 1. Then we have phosphates over here linking to position 5. If we have 3 phosphate groups, it's a triphosphate, hence the tri. If we lose one, we have a diphosphate because we'd only have 2 left. And if we only had 1 phosphate, it would be a monophosphate. These are the building blocks of a nucleotide. When we look in a little more detail, we can compare the sugars of DNA or RNA and see that there's really very little difference, but it's an important chemical difference. Ribose has an extra oxygen, that's at position 2. We have 1 more oxygen there compared to DNA, or the deoxyribose sugar in DNA. When we lose that oxygen at position 2, we have deoxyribose. Deoxy means missing an oxygen. Ribose is an RNA sugar. Deoxyribose is the DNA sugar. Waving off of position 1, we're going to have one of the bases. In DNA, we have the A, the C, the G, and the T. But in RNA, we're going to have to swap the uracil, U, for the thymine, the T, that you find only in DNA. Here, look at the small difference in the structure of uracil and thymine. We have this little chemical difference only in one position. We have a hydrogen in the RNA, and we have a methyl group in the thymine, a carbon with 3 hydrogens. Remember, that's a methyl group. Those are the only chemical differences in DNA versus RNA. The oxygen or the lack thereof in position 2, and which we designate as ribose or deoxyribose, and the thymine found in DNA and the uracil found in RNA. That's really it, but those chemical differences do cause differences in the functionality. Let's go back and take a look where the bases are located on the nucleotide. So where are those bases? They're overhanging out as a kind of flag on the carbon position in one of the sugars. Then on the opposite side of the nucleotide, at the carbon sticking up, the sort of arm, this is where the phosphates are linked in the nucleotide. So that's the basic nucleotide structure. Commit the general position information to memory. I'm going to draw it out again. This is 1, 2, 3, 4, 5. Phosphate, 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 base. So now we'll do a quick overview of the DNA. For some, this is a review, but for others, it may be new. I wanted to show you how we have the bonding between the phosphates and the sugar so that the sugar phosphate backbone of the nucleic acid and the sugar phosphate backbone is bonded covalently. So that's a much stronger bond than what you would find between the bases, between the two strands. And the strands are labeled as 5' and 3'. What exactly is that referring to? Well, it has to do with the carbons in the sugar. If you look at the orientation of the carbons in the sugar, which we just talked about a minute ago, 1, 2, 3, 4, 5, the 5' end has the number 5 carbons sticking out, and the 3' end has the number 3 carbon sticking out. So that's what the 5' or 3' orientation is talking about. And in order for the bases to be able to hydrogen bond to each other, the two strands have to be running in opposite directions. That's termed anti-parallel, so that the bases can face each other to hydrogen bond. So if one strand over here is 5', then the other strand must be 3'. They're running opposite each other. That's anti-parallel. Let's look again carefully at the nucleotide. So looking at the nucleotides, now sort of flipped in the other direction compared to before, we have the nitrogenous base over here at position 1, and then at position 5, we have the phosphates. And how do we refer to these things? Again, we're looking at an RNA nucleotide, and we would refer this as adenosine triphosphate. But if you got rid of one of the phosphates, you're left with 2. It would be a diphosphate. And if you got rid of 2, you'd be left with only one phosphate attached, and it would be monophosphate, adenosine monophosphate. And if you get rid of all of the phosphates, only looking at the ribose and the nitrogenous base, then it's referred to as a nucleoside. So that's the terminology listed over here. If you're confused and you're trying to figure out what the book and other sources are talking about when using all of this different terminology, hopefully this little diagram from your book can help clarify some of that for you. I like this diagram for that reason because it's not always very clear. So this is the end of the chemistry chapter. Phew. That was a lot, right? But it's an important chapter, and if anything is unclear to you, you should first come back and listen to just the sections that were confusing. There are some extra recordings on some topics posted for you as well. I wanted to point out in one of the first opportunities of the semester that you have some extra credit possible. You can only do so much extra credit in the class before you max out. This is one. Don't bother with it if DNA polymerization is something that you have covered in depth before. There are other opportunities you can take. But if you find DNA polymerization a little bit confusing to you, you can do the constructed DNA molecule directed learning activity, abbreviated DLA from here on out. And for this particular exercise, you're going to have to do some coloring, cutting, and taping or pasting on paper. This work will force you to visualize how the molecule is put together, which parts of the molecules fit together, and the anti-parallel nature. So if this is semi-unfamiliar set of concepts for you, then go ahead and do it, and it'll help clarify. If you have this material down, you don't need to do it, as other opportunities will be coming up that you might want to spend your time on. So that's it. Thank goodness. We're done exploring the chemistry of biology. I'll see you in chapter 3.